So you may have heard the term gene therapy in the news. Well, here we're going to learn a little bit about that process and how it's been used. So first off, what is gene therapy in general? Well, gene therapy introduces healthy genes into cells that lack them. So it was first used successfully in the year 1990. Two girls were cured of a rare blood disorder caused by a defective gene, and the girls remained healthy, even well beyond that. So what this is in the most basic form is kind of repairing genes or fixing a broken gene. It's finding and identifying a region or a gene that is defective, identifying that area and replacing it with a healthy gene and allowing that cell then to replicate, causing that now normal or healthy gene to proliferate in the cells. However, this has other cases and implications. One example would be cloning of animals. So here's just an example of Dolly the sheep. How is Dolly the sheep clone produced? Well, it's through this kind of concept of gene therapy. Now, the successful embryos, about 30 in 277 tries, so again, not a great percentage, were transplanted into a surrogate mother sheep. What did this process look like? So even though only 1 in 270 tries was successful, in July 5th, 1996, Dolly was born. How did that process go about? Well, you take that donor cell from a sheep, and we extract the nucleus is extracted from that. So in this case, only the nucleus is being used. We have this egg cell taken from an adult female sheep, and that nucleus of the egg cell is removed. So essentially from this sheep, we have the egg cell components and machinery, and from this sheep, we only have the nucleus. Through an electrical shock process, that nucleus is transferred into that fused egg cell. That egg cell then is induced to divide normally. That single fused egg cell will then produce an embryo. That embryo will be placed into a foster mother, and that foster mother will ultimately have the cloned sheep as a offspring. So the embryo develops normally, and that baby lamb is produced. In this case, it was Dolly. So since Dolly, scientists have successfully cloned sheep, mice, cattle, goats, and pigs. However, problems and complications still arise, leading to premature death. Not really sure why that's the case, but Dolly died in 2002, having lived only half a normal sheep's life. So while the process does make sense that we're just taking the nucleus from one and the egg cell from another, there's still a potential for shortened life expectancies. Now, Yukon has gone through with large animals, and in 1999, the birth of the first cloned calf from an adult farm animal in non-reproductive cells in the United States. Its name was Amy, and Amy has given birth to a 103-pound calf in 2002. Now, if we look here, this is kind of the mother, if you will. These are the clones. You can see that they also have a very similar kind of white triangle on the top of their head there. And these were all the clones. And I forget all their names, but it was A, B, C, D, and E. They had the name that came with letter A, B, C, D, and E, and this was at UConn. So stem cell basics, how is all this possible? Well, it's through the process of stem cells. So stem cells have a very unique characteristic to them. They're what we call pluripotent, and that means they're capable of forming an entire organism. One cell can form basically all the components of an organism. As development proceeds, cells lose their pluripotency, so they become committed to one type of tissue. And these are called adult stem cells because they still have the genetics to produce anything, but they themselves kind of become specialized and only produce one particular type of cell. So since you arise from just one cell that had to have the ability to perform and produce all these different tissue types. However, as you get older, heart muscle cells tend to produce more heart muscle cells, and neurons for the at least first 2 years, they can produce more neurons. Intestinal cells produce more intestinal cells. They become more specialized. They start to lose that pluripotentness that you originally had when you were very, very young. Now, when we're looking at stem cells, there's some benefits and some drawbacks. So benefits, embryonic stem cells could be used to restore tissues lost or damaged due to accident or disease. Experiments have already been tried and successful in mice. Damaged spinal nerves, as I mentioned, nerve cells don't divide for very long into your life. So damaged spinal nerves can be partially repaired, can help someone recover from a spinal cord injury, might be paralyzed in parts. So that's some of the benefits to stem cells. Some of the problems, though, is finding a source, especially for those embryonic stem cells. Harvesting them from discarded embryos raises ethical issues. Are you killing unborn babies? And then there's also the possibility of an immune system rejecting them. So implanted stem cells will likely be rejected by the immune system of the individual. So it's important to realize some of the benefits, but also be mindful of some of the issues or problems when you're making a decision going forward. These are things that science has to do, is constantly balances the pros with the cons.